My dear students, today we'll be talking about one of the important topics, that is double fertilization in angiosperms. And this is the revision short 12. This double fertilization is unique to the angiosperms. You know that flowering plants, double fertilization happens. Two fertilization events in one embryo sac. That's why it's called a double fertilization. Number one, you know that when the pollen pistil interaction happens, that means this pollen grain it germinates on on this stigma of the female plant, or stigma of the female flower, you can say, and the pollen tube is generated. That pollen through that pollen tube, these generative cell nucleus, two of them, they enter into the embryo sac. Now, what is guiding this pollen tube into the embryo sac is the synergid. So one of the synergid is actually degenerated once the pollen tube enters into it, and the release of these two generative cell nuclei, they are released into the pollen tube itself. Then after that, one of the this generative cell nucleus, it fuses with the egg cell and forms the zygote 2N, and this process is called syngamy, and then zygote results into the embryo. Then after that, triple fusion happens. That's male gamete. One of the male gametes, it fuses with the two polar nuclei of the central cell and it forms PEN, that is primary endosperm nucleus. Now, this process is called a double fertilization. Why? Because one of the generative cell nucleus, it fused with the egg cell. That fertilization happened. That's called a syngamy, and in triple fusion, another generative cell nucleus, it fused with the polar nuclei of the central cell. So these two fertilizations are happening at the same time. That's why it's called double fertilization, and it is unique to the plants, flowering plants only. Now, after post-fertilization, this pen turns into the endosperm and zygote turns into the embryo. You can see in this diagram, it's given in your textbook, it is showing the degeneration of the antipodals. Antipodal cells are degenerating, which are towards the clasal side, and these synergids are also degenerating, which are towards the micropyle end. And now, what was the role of these antipodals? Though it's not given in your textbook, but you should still know what's the role of the antipodals. Number one thing, that is they have the nutritive role. Antipodals help in transport and metabolism of nutrients from the nucleus to the embryo sac. It is important in cereals like wheat, rice, where antipodals are large and persistent. They have physiological support also. They provide physiological support also. They maintain embryo sac metabolism. They help in maintaining osmotic balance and cellular signaling. So when these antipodals and synergids, they are degenerated, what happens to their material? Please remember that the material that comes from the degeneration of synergids and antipodals, it is reabsorbed locally by central cell, developing endosperm. These nutrients which are there, like proteins, lipids, nucleic acids, they are broken down and they are used for rapid endosperm development. Now, how do these cells degenerate? There's a process which we call it PCD. We call it as PCD, that is programmed cell death. Programmed cell death is a genetically controlled process. It's not accidental death. So it is initiated by the body for the degeneration of these synergids and antibodies. Then how does this start? What tells these antibodies that they have to degenerate, or what tells these synergids they have to degenerate? So there are signals which are responsible for degeneration. Like, for example, when fertilization happens, the fertilization triggers the signals, like when the entry of the pollen tube happens into the this synergids, and release of the male gamete happens, then syngamy and triple fusion happens. These chemical events, they actually trigger the degeneration of these synergids and the antipodals. And there's one more thing: there is a rise in the auxin and cytokinin level at the time of these endosperm formation. So these are the signals which are actually telling these synergids and antipodals to degenerate. So this is about double fertilization. I hope you understood the whole process. Please wait for the next video. Thank